Hello everyone. So in the previous lecture we have seen the digestion of carbohydrates, and during digestion we have produced the monosaccharide units. That is, in the digestion process you have seen that we have got glucose, we have got galactose and fructose mainly during the digestion process, and these monosaccharide units are present in the jejunum area where the absorption usually occurs. See, the absorption process of glucose, galactose and fructose, they are a bit different. Okay, the glucose, galactose and fructose, these are hexoses. Hexoses have different mechanism of absorption, whereas pentoses have a different mechanism of action. Okay, these glucose and galactose are absorbed by the same mechanism. These glucose and galactose, these both are absorbed by the same mechanism, which is known as sodium dependent secondary active transport. It is absorbed by sodium dependent secondary active transport, also known as SGLT, whereas fructose is absorbed by facilitated diffusion. Okay, now if you see the absorption of glucose and galactose from the intestine, you see, suppose that this is your intestinal lumen where the absorption is usually occurring. Suppose this is your intestinal lumen, this is your lumen, and this is your intestinal epithelial cell. Okay, these SGLTs are located in the apical part of the intestinal epithelial cell. So this is your apical part where these SGLTs are located. SGLTs, or sodium dependent glucose transporters, specifically SGLT1. SGLT1 is located in the apical part of the intestinal epithelial cell from where glucose and galactose are being absorbed in the intestinal epithelial cell. It is called sodium dependent glucose transporter or sodium dependent secondary active transport because it requires sodium. Along with sodium, glucose or galactose can be absorbed in the intestinal epithelial cell. Okay, so if you see in this lumen, this is your lumen, glucose is present, galactose is present, and fructose is present. So glucose and galactose can be absorbed via this SGLT1. This is glucose, can be absorbed from the apical part of the intestinal epithelial cell inside the cell along with sodium. Along with sodium, this get absorbed. Okay, now this absorption of sodium is actually facilitated by a pump which is known as sodium potassium ATPase pump, which is located in the basolateral side of the intestinal epithelial cell. So this is your sodium potassium, suppose this is your sodium potassium pump which is located in the basolateral side. This is your sodium potassium ATPase pump which is located in the basolateral side of the intestinal cell. This is your basolateral side. What this pump will do, what this sodium potassium ATPase pump will do, that it will pump 3 sodium outside and it will take 2 potassium ions inside the intestinal epithelial cell. Now when it pumps the 3 sodium ions outside, the concentration of sodium inside the intestinal epithelial cell will decrease. So the concentration of sodium inside the intestinal epithelial cell will decrease. So due to the difference in the concentration gradient, the sodium from the lumen, the sodium which is present in the lumen, intestinal lumen, it will move to the inside of the intestinal epithelial cell, and along with the sodium, the glucose will also move inside the intestinal epithelial cell from the lumen. So you can see that here the ATP is being used, because during this sodium potassium ATPase pump, and during when the 3 sodiums are going outside and 2 potassiums are coming inside, ATP is used. This ATP gets converted to ADP. So energy is used here. Energy is used here, and due to the use of this energy and due to pumping in and out of sodium and potassium, there is a concentration gradient of sodium is created, and due to the difference in the concentration gradient, the sodium from the lumen moves inside the epithelial cell. So you can say that the movement of sodium and glucose inside the intestinal epithelial cell is using the ATP indirectly. If there is no ATP used in the potassium ATPase pump, then it would be difficult for the glucose and sodium to move inside. So that is why this SGLT is also known as sodium dependent secondary active transport, because it is using sodium and ATP is being used indirectly. Has it been used directly, ATP used, so we would have called it a primary active transport, but the ATP is used secondarily, so we are calling it sodium dependent secondary active transport. I think it might be clear to you till now. Now the glucose has come inside to the intestinal epithelial cell. Now absorption, if you are talking about absorption, glucose has to be traveled up till the bloodstream. So suppose this is your capillaries nearby the intestinal lumen or intestinal cells. There are special transport which are known as gluts, which are located in the basolateral side of the intestinal epithelial cell. So here are the gluts, which are known as glucose transporters. This is GLUT, glucose transporter. In this case, in the intestinal cell, we have GLUT2. With the help of this GLUT2, the glucose travels from the intestinal epithelial cell into the capillaries, and after entering the capillaries, it is distributed through the portal vein, it is distributed to the whole body. So that is how glucose is being absorbed. Same mechanism is being carried out by galactose also. So galactose also uses this sodium gradient. So galactose can also come, and galactose in the basolateral side of the membrane, it can either use glucose transporter 2 like this one, or it can use another glucose transporters which is GLUT5. Sometimes, mostly it uses GLUT2, but this galactose can also be transported by GLUT5 also. Okay, so glucose and galactose are absorbed by this mechanisms. Okay. Now talking about fructose, it is being absorbed by facilitated diffusion. This fructose doesn't use any sodium gradient, it doesn't use. It simply gets transported by facilitated diffusion. So to for absorption of fructose, specific glucose transporters are used, which are known as GLUT5, which is also located in the apical part of the intestinal epithelial cell and also the basolateral side of the intestinal epithelial cell. These light blue ones are the GLUT5s. Through this GLUT5, fructose is absorbed by facilitated diffusion. And after entering the intestinal epithelial cell, it gets absorbed into the capillaries. Clear? So this fructose is not making any use of any sodium or indirect ATP, whereas it is being absorbed through facilitated diffusion directly into the bloodstream, whereas pentoses are absorbed by simple diffusion. Pentoses are absorbed by simple diffusion. It doesn't need any gradient, doesn't need any transporter. It simply gets absorbed by simple diffusion. Okay. I think that would be all for today's lecture. In the next lecture, we will talk about various glucose transporters. In this lecture, you have seen few glucose transporters like SGLTs and GLUT2 and GLUT5. Apart from these glucose transporters, there are other transporters also. We will see that in the next lecture. Okay, thank you.